Hello everyone, my name is Stefano Ciavatta and I am a senior scientist at the Plymouth Marine Laboratory and at the UK National Centre for Earth Observation. I'm also co-chair of the Marine Ecosystem Analysis and Prediction Task Team of Ocean Predict, which is an international organization promoting the development and exploitation of operational oceanography. Today, I'm going to talk to you about the assimilation of biogeochemical data in marine ecosystem models, providing a review of the state of the art and some ideas for future developments. Data assimilation is something that we exploit daily. Just think to the meteorological forecasting centers that predict a sunny summer day for tomorrow, but then they observe snow at midday, so they will integrate the new data, they will assimilate the new data in their models to provide a better forecast in the afternoon. In practice, data assimilation improves the weather forecast that we watch every day. This is something that is happening also in modern ocean operational systems that provides predictions of variables of the ocean such as water temperature or chlorophyll concentration. Chlorophyll concentration, which is an estimate of the biomass of phytoplankton. Phytoplankton are the small algae that are floating in the ocean and that are so relevant to us because they are at the base of the marine food web. They provide oxygen in the atmosphere, 50% of the oxygen that we breathe. And they also assimilate the carbon dioxide, bringing it from the atmosphere to the deep ocean via the biological carbon pump. But besides phytoplankton, operational centers provide predictions also of other variables like nitrate, oxygen, pH, which are all relevant, for example, to achieve the Sustainable Development Goals. And operational centers to provide predictions on these biogeochemical variables use assimilation of ocean color very often. But is the assimilation of ocean color useful? Yes, it is. Here we have an example from the Operational Center in the Northwest Shelf that assimilated data of ocean color into their model to improve the simulation of phytoplankton chlorophyll. We see that the blue continuous line assimilating the data matched closer the ocean color data, the yellow line, than the model without any data assimilation, the dotted blue line. Importantly, the assimilative simulation improved also the simulation of the in situ data of chlorophyll that were not assimilated. But what about the simulation of nitrate? But actually, the simulation of ocean color had mixed results on nitrate. It improved the simulation in the winter months, you see the blue line closer to the black dots, but it deteriorated the model simulation in summer. You see the dotted line is closer to the data in summer. And the idea is that we will explore today is why not using biogeochemical algal floats data assimilation to improve the simulation of nitrate, and we will see also other examples. But first, let's see what are these biogeochemical models we are talking about. Here we have a representation of a quite simple ones that connect the ocean states, phytoplankton, nutrients, zooplankton, and so on, by means of arrows. These arrows are actually mathematical equations that describe ecosystem processes, for example, photosynthesis, for which we have oxygen produced by phytoplankton. And oxygen in this case become a state variable of the system. And the equation includes also a model parameter, which is the maximum rate of phytoplankton production of oxygen. These biogeochemical models are typically coupled to physical models that provide transport in the horizontal and in the vertical, and also provides inputs like temperature, salinity, radiance. Then the outputs of these coupled models can be compared to data from ocean color, for example, phytoplankton chlorophyll, or for in situ systems like gliders or biogeochemical algal. And what if we decide to assimilate this data into our model? But we have an operational model that provides a forecast of chlorophyll. This forecast will have an error because the models are uncertain. We get a data of chlorophyll. And the truth, the true value of chlorophyll in the system will be hopefully somewhere in the between, but we don't know where. So we apply data assimilation, which balances the forecast and the data and the error to get closer to this true value of chlorophyll in the system. The analysis value will be used to initialize a new forecast. Two things are important to note at this point in this talk. But firstly, we can somehow improve also variables that are linked to the variable that we are assimilating. For example, oxygen, even if we are not assimilating it. And we can also use assimilation to improve the estimation of biogeochemical model parameters. For example, the oxygen production rate that we have seen before. And another important note is that the true value of chlorophyll, we don't know it, what is that in the real system. And sometimes we can pretend to estimate it via independent datasets. Or in twin modeling experiments, in theoretical exercises, we can use a model to produce the true value, to produce data, to assimilate this data into the model for theoretical exercise of sensitivity of the system to different assimilation strategies. Okay, this was the introduction, but what are the key questions that we are addressing here today? Is the assimilation of physical data sufficient to improve the understanding and prediction of marine ecosystems? And for the same objective, is the assimilation of ocean color data enough? Is the planned number of biogeochemical cargo floats, 1,000, sufficient? And then at the end, I will give a vision for a fully integrated and smart global ocean observing system. But first, the first question, is the assimilation of physical data sufficient? No, unfortunately, it is not. In fact, this was demonstrated by Park and Kotos in 2018. Here we have a picture from their paper where we have data of nitrate and a model simulation of nitrate in the global surface ocean. We see that the two match pretty well. Red matches the red and blue matches the blue. But this is deteriorated when we assimilated physical data. You see that yellow patch that is not observed in the data. So physical data deteriorated the simulation of nitrate. Why did it happen? But they looked at the vertical velocity that were simulated at the equator with the model and with the model with data simulation. Here they are, the vertical velocities in the model left and after data simulation on the right. On the right we see that there are anomalous highly values of vertical velocity that were triggered by the simulation of the physical data. As a consequence of these high values, nitrate was upwelled from the deep ocean into the surface where it appeared with that patch that was not represented in the data. So why this is happening and how to improve physical data simulation in this context is under investigation. But here today I will discuss if the assimilation of biogeochemical data from floats can help to fix this issue and maybe constrain physical simulations. But the idea is inspired by the work of Yu and Kotos in 2018, that they did some twin modeling experiment with an ideal ocean channel. So in practice, they used a coupled physical biogeochemical model to generate true value of temperature and nitrate. They extracted a synthetic observation from this simulation and they assimilated them into the model and compared different assimilation strategies. So here are the true value of temperature and nitrate simulated by the model. We see that there is a clear anti-correlation between the two. Temperature is high at the surface where nitrate is low. Nitrate is high at the bottom where temperature is low. And in fact, if we scatter plot temperature and nitrate from the model simulation without data assimilation, we see that there is a clear anti-correlation between the two. So the authors extracted physical biogeochemical pseudo-observation from their nature run, and they did some trials of assimilating these variables into the model and looking at what happened to different parts of the model. Firstly, they assimilated the physical data to correct physics only, and this led to a deterioration of the correspondence between the temperature and nitrate, as shown by the larger dispersion of the points in this scatter plot. Then they assimilated physical data to correct both physics and biogeochemistry, and we saw that these helped to preserve the correspondence between temperature and nitrate. But they obtained even better results when they assimilated biogeochemical profiles to correct both physics and biogeochemistry. So the idea now is clear. Why we don't assimilate biogeochemical Argo profiles to help to constrain the physical simulations and to preserve the consistency between physics and biogeochemistry? This is an idea that for regional seas is being explored by a European project Seamless. Is the assimilation of ocean color data sufficient? Unfortunately, it is not, because ocean color assimilation is very useful. As you know, ocean color radiometry measures the fraction of the solar radiation that, after entering the ocean, interacting with the particles, return out of space. And one of the most important particles that interact with the solar radiation is phytoplankton, which uses it for photosynthesis. And the satellites can see up to where the solar radiation can go in depth. And this is normally in the order of the tens of meters, not in the deep ocean. Anyway, ocean color assimilation has been used for the product of total chlorophyll in the seminal work of Ishizaka in 1990, and after that other products from ocean color has been assimilated, particular organic carbon, spectral attenuation coefficient, remote sensing reflectance, but also phytoplankton functional types. And some of this research was pulled through the operational systems. For example, the Northwest European Shelf Operational System assimilate phytoplankton functional types to investigate, to predict the distribution of the functional groups and study the composition of the phytoplankton community. So there is clear advantages of the use of ocean color assimilation because we have global coverages with daily frequency of a very important component of the global ocean. But there are also some drawbacks. With ocean color assimilation, we can constrain mainly the surface layers of the ocean biogeochemistry. And so the idea is, okay, let's complement this positive work of ocean color with the assimilation of biogeochemical Argo profiles in the deep ocean. And this has been done, for example, by Wong and co-authors in recently that optimized the parameters of biogeochemical model by using different data sets collected in the Gulf of Mexico, represented here. So here we have trajectories of a number of biogeochemical Argo flows and the dots that represent where they applied the 1D model. They use the 1D model to simulate chlorophyll, phytoplankton biomass, and particular organic carbon along the water column, and they compared it to the data from the biogeochemical Argo flows averaged. And we see that there is a good match for chlorophyll, phytoplankton biomass, but not for particular organic carbon, which was definitely overestimated by the model at the surface. So they thought, okay, let's use surface ocean color data to better parameterize the model. And they did it. And they, in fact, improved the simulation of the variables at the surface. But at the same time, they deteriorated the simulation in the deeper layers because they were not able to constrain the simulation by using the surface chlorophyll data. So they used also the profiles of chlorophyll from the biogeochemical Argo float. And in this case, yellow line, they managed to catch better the chlorophyll profile that was observed. But at the same time, they deteriorated the simulation of the phytoplankton biomass and the particular organic carbon biomass. And that's because the chlorophyll data alone cannot really help that much to optimize the simulation of the phytoplankton biomass when you are using a variable chlorophyll to carbon parameter in your model. So what they did, they added on top the use of data of phytoplankton biomass and POC from the biogeochemical Argo. And blue line, in this case, they optimize parameters that led to a good simulation of the observed observations. But perhaps even more interestingly, they managed to improve the biogeochemical model by assimilating the biogeochemical algal flow data. And they also estimated the merging property of the system. That means they managed to estimate better the rate of sinking of the particle organic carbon, and also as a consequence to improve the prediction and the simulation, of the fraction of primary production that was exported in the deep ocean as particle organic carbon. So assimilation of biogeochemical algal to improve the model and to estimate on merging properties of the system. So, is the planned number of biogeochemical algal flows sufficient? But this was investigated by Ford and co-authors without co-authors, by means of a twin modeling experiment in which they used a global coupled physical biogeochemical model to generate true values of biogeochemistry in the global ocean. Then they generated 1,000 biogeochemical algal flow datasets of chlorophyll, nitrate, oxygen, and pH in correspondence of as many 1,000 algal flows that were deployed in the real ocean at the moment. And then they assimilated these datasets into an alternative model of the global ocean and looked if these managed to improve the results of assimilating ocean color only. And yes, they did. They improved the simulation obtained from ocean color assimilation by assimilating 1,000 biogeochemical algal flows. Here we have the image of nitrate simulation at the depth of 100 meters for just 1 month, and the blue shows where the assimilation of the biogeochemical algal flows improved the simulation that was assimilating ocean color only. We see that there is a large dominance of blue. The assimilation of the algal flows constrain and improve the simulation of nitrate. Finally, I would like to give you a vision for a fully integrated, smart and global ocean observing system to improve the understanding and prediction of marine ecosystem. A vision that is inspired by what is happening at the moment now on my back in the English Channel, where we have a glider which is driven by the operational predictions of chlorophyll given by the model run at the Met Office. And in practice, what is happening is that we have a glider that is measuring chlorophyll. These data are transmitted to the operational center that assimilate the data along with ocean color and provides forecast at the 7-kilometer resolutions. These predictions are downscaled to 100-meter forecasts of chlorophyll, and these forecasts are given to the pilot to redirect the glider towards the maximum of chlorophyll that has been predicted. Why we are doing this? But to study the small-scale variability of biogeochemistry that lead to the phytoplankton blooms, but also as a proof of concept if we can use gliders to detect gliders and models to detect in advance harmful algae blooms, but also to find a system to optimize monitoring network using less gliders because models are driving them where it's important them to be. The idea is that we don't apply the same philosophy with the biogeochemical algal fleet. They can collect chlorophyll data, transmit them to a global ocean forecasting center where they are assimilated to provide better forecasts of the chlorophyll distributions. And these better forecasts are used to reprogram the frequency of the descent-ascent cycle of the glider and the frequency of the vertical measurements. And why to do that? But to early detect phytoplankton blooms, which are relevant for fisheries, for example, but also to study the variability of biological carbon pump. If we are predicting that there is a bloom somewhere, we can reprogram the glider to measure with a higher frequency the particular organic carbon that is sinking as a consequence of the bloom. but also apply this merging of biogeochemical Argo and models to optimize the energy consumption of the biogeochemical Argo fleet. We make it work more frequently when we predict something is happening, and we can keep it quiet when the model predicts that nothing is happening. And the picture actually is completed by the use of ocean color from satellite for assimilation and do better predictions, along with gliders that are driven cleverly by the model prediction and provide the data that are assimilated into the model to explore, for example, the blooming on the horizontal dimension. But here I've been talking about studying the phytoplankton bloom, but you can imagine other applications of such an integrated system. For example, to detect and investigate the shallowing of the oxygen dead zone in the global ocean, as an example. And finally, my recommendations. So I would suggest to exploit biogeochemical Argo flows to improve biogeochemical models, to improve their parameterization, and also to investigate emerging properties of the system along with the models. I gave a vision for a fully integrated global ocean observing system that really merges Argo gliders with models to get better prediction of the global ocean biogeochemistry. Towards this vision, it's important to use biogeochemical flow data assimilation to complement the satellite data assimilation that can see just at the surface. And I would test if the assimilation of the biogeochemical Argo data can help to constrain physical variables, or at least to help the physical data assimilation in those parts of the ocean where physical data assimilation deteriorates biogeochemical simulations. For example, in the equator, we have seen an application. And we have seen that 1,000 biogeochemical data, biogeochemical flows had a significant impact on biogeochemical models, at least in twin experiment. Let's verify it really with a real deployment of these Argo flows. And then, okay, I didn't touch at all today on the need of evolve the biogeochemical data assimilation methods, stochastic ensemble, non-Gaussianity, and the use of artificial intelligence and machine learning. But all these are topics for another topic, another day, another talk. Just the time to acknowledge the persons that kindly helped me with this presentation, Katja Fennel from the Deleuze University, who is also co-chair of the Marine Ecosystem Analysis and Prediction Task Team of Ocean Predict, Gianmario Rinaldi and Shannon Grossberg of the University of Exeter, David Ford and Susan Kane from Met Office, Giorgio Dall'Olmo and Joseph Scacala from the Plymouth Marine Laboratory. Some of the results that I've shown today comes from the project Campus, which was funded by the UK NERC. And I acknowledge the support of the Copernicus Marine Service that through the Service Evolution funding stream funded the project Tosca and Optima that inspired some of the ideas presented here. And then some of the ideas that were presented are going to be developed in the European project Seamless. At the end, I thank you for your attention, and please let me have somehow your questions. Have a good day, and bye.